So now it's my honor to introduce our first keynote speaker, Dr. Rosemary Gillespie, who is a professor and Schlinger Chair in Systematic Entomology at UC Berkeley, where she's at the Essex Museum of Entomology and the Department of Environmental Science, Policy and Management. She's past president of the International Biogeography Society and the American Arachnological Society. She got her BS from Edinburgh, PhD from University of Tennessee, and was on faculty for a number of years at University of Hawaii before moving on to UC Berkeley. Dr. Gillespie's work has made fundamental contributions to our understanding of speciation, adaptive radiation, island biogeography, island conservation, and of course, community assembly. And indeed, she's made major contributions to our understanding of the evolution of the ecological communities, particularly through pioneering studies in ocean archipelagos. Her work's been published in a number of high-profile papers that have become classics in the field, as well as book chapters in some of the most important compendia on island biogeography and adaptive radiation. Her fieldwork has also advanced our understanding of arthropod distribution, diversity, and natural history through field surveys on many island groups, including Hawaii, Fiji, Micronesia, French Polynesia, and the Mascarenes. And most recently, she's involved in an NSF grant, a collaborative grant, which is a biotic inventory of terrestrial vertebrates, spiders, and Hemosporidian parasites of Indonesia. So we're honored that Dr. Gillespie is here to be our keynote speaker, and please join me in welcoming her. On the panel.
Thanks so much. It's a tremendous pleasure to be here and a real honor to be invited to this event. What I want to do today is talk to you about the work that we've been doing on islands, and I'll be focusing in Hawaii and focusing mostly on spiders. And so what I want to do, if I can get the hang of things here. It's quite up there. So I'm going to be talking to you about very isolated systems here. And so when we think about these island systems, I'm going to be focusing on ones that are really way on out there. And so you're talking about extreme isolation. And if you think of over short periods of time and a very isolated system, nothing happens. But if you have an island that has sufficient area and sufficient isolation, these are conditions in which you're likely to get speciation. So here as you get more time, even on the most isolated archipelago is going to get propagules. And over time, if there's not so much immigration into that system, you'll tend to get speciation. And so at the most, with the longest time and the greatest isolation, this gives organisms a lot of time to explore ecological space, and these are the conditions under which you tend to get adaptive diversification. And so what I'm going to focus on in the course of this talk is the Hawaiian island system in particular. It's the most isolated archipelago in the world, so it's very discrete, and so you can see things, see how things actually diversify within this context. So first of all, just a little bit of background, just so that you can get a feel for what is going on in Hawaii. We hear so much about Hawaii's so well studied. Everyone knows about Hawaii, and I want to just give you, kind of bring us down to ground as to exactly what has happened there and why, and what has not, in particular, what has not happened. So just a little bit of background then. So first of all, when we think about Hawaii, we often think about, the beaches, the palm trees, the tropical waters. But what I'm talking about here is the high elevation systems where you get most of the endemic species, as in many other of these kinds of systems. So in Hawaii, you get the cloud forests at elevations above about about 2,000 feet or so. And it's in these kinds of environments where you get very rich diversity and very high levels of endemism. So when, in terms of what's known about these systems, really, when I first came to Hawaii, this is really where we were at. That we were relying, and we're still relying heavily on work that was done in 1900. And this is a work by R.C.L. Perkins, who was a naturalist who came to Hawaii in the late 1890s. And so he wrote up the Fauna Hawaiiensis, the Fauna of Hawaii, in 1900 and subsequent years. And he single-handedly went throughout the Hawaiian Islands collecting. And you can see here that some of the, this is just a quote, I can't read it like from there, but I was obliged to carry a limited supply of oil and a small stove on account of the difficulty or impossibility of making a fire. And this is because of the continual relentless rain in these environments. It's raining constantly, really, and you know, so his end thing was, this will be as much as a collector will care to carry in so rough a country. And I found the oil and stove a sore burden to carry. So you can see that this was not, that they're not the easiest environments to be working in. But just to, the thing is, when Perkins was there, though, he was actually, the insights he gained from these systems was really quite extraordinary. And he made some of the key observations in Hawaiian Drosophila in particular. So here you see that some of the behaviors that even he was observing at that time. And so he talked about how species were added to the system. And he talked about how evolution was happening. And if you think, this was in the 1890s. And so he was able to really figure out some fine details through his observations, some really extraordinary details about the biology of these systems. And I should say it was based on Perkins's work, and in particular on flies and other insects, that some of the early work on Hawaiian flies was really made possible. And this is work that by Hamp Carson, and I'll come back to Hamp Carson's work later on. But Hamp Carson was a geneticist at the University of Hawaii, and he started the Hawaiian Drosophila project. And he built this tree here that was based on the lengths of the chromosomes of these Hawaiian Drosophila. He used the fact that the Drosophila have these giant chromosomes and was able to piece together the relationships between them based just on the morphology of these chromosomes. So this was really the first phylogeny that was produced for any Hawaiian organism. But then we think, you know, what was actually going on during that time in other groups, you know, this is, you know, through the early part of this century, of the last century now. So during that time, there was actually very little natural history on the Hawaiian arthropod fauna until really the 1970s and 1980s. That's when there was a kind of renaissance in understanding the arthropod biodiversity. And really through three people, this is Frank Howarth, Wayne Gagne, and Steve Montgomery, were the three that really kind of started up the understanding of Hawaiian arthropod biology again. And the important thing here was that they went out into the field and actually collected organisms and really did the natural history that was really necessary to provide the foundation for other biologists to come along and do the work. This is just a Eupathecia caterpillar, and if you just wait in anticipation here, you see this is actually you see what it does. It's a predatory caterpillar, and it was Steve Montgomery through his observations that he figured this out. So this was really remarkably recently, where you know things were just beginning to be looked at. And this is what it was like when I first arrived in Hawaii, which was in 1987. And what the thing here is that, you know, I've been talking about going up into the mountains. And when I got to Hawaii, actually, there was really very little work that was being done in these systems. And so when I got there, I found these spiders. They're unknown, undescribed, so exciting. And so I wanted to keep looking at them. And the thing in particular about these spiders is that they are just sitting there. They're mostly nocturnal. This one happened to be out in the day because it had got a prey item. But they don't see. They just sit there, and you can really work with them. You can watch what they're feeding on. You can watch where they build their webs. You can observe their behavior, see what they do, where they like to live, why they like to live, where they like to live. And so they're very good organisms for trying to understand the interaction between the environment and the morphology of the organism. And also how it diversifies. But the thing is, what we're talking about here is that we're talking about high elevation forests. It's really tough to get to, and not many people working there. And so how do you actually work in these systems? And just to give you a feel for how things started with me, you know, here I was stuck. I wanted to get up into these environments, and who do you take to go with you? Who's going to stick with it, you know, in that rain? So what do? You get your mom. So I brought my mom over, and she stayed with it for a while, but she's from Scotland, you know, and it's a long haul from Scotland to Hawaii. So anyway, I found someone else who stuck with it as well, and so we got married so that it stuck even harder. And then the hope was that the F1 might be just as useful, but they were of limited use, at least for a while. This is how basically this is how things started with me. But I think the point I want to make here is that the most fundamental thing in starting all of this work that I'll be talking about is to know the system really well. And we've heard this before from many others that we've learned from. Bob Ricklefs will argue this a lot, and many others as well. But this is a quote from Jim Brown that I like particularly much. So this is, he said, early on, George Bartholomew told me that you need to have one home taxon to really understand ecology. I'd now add that you need one home region as well. If you know one taxon, one region really well, you begin to see patterns in the rest of the world. These systems that you're familiar with give you the confidence to work on the organisms and places and give you insights into how to begin testing the broader patterns. So I think for anyone starting out, I think this is a really important fundamental piece of the puzzle to have under your belt before you get going. So the system then that I've chosen and that we'll be talking about here are isolated hotspot islands. And as I said, I'll be focusing in Hawaii. And the Hawaiian system is a hotspot, as are many of the islands through the Pacific. So basically, you've got the mantle plume that just churns up lava and forms islands that just pop up to the surface. And so what happens is you just get this continual formation of islands. If the tectonic plate did not move, you'd have an enormous island there at the moment. But the thing is, it does move, and so because of that, you've got these little islands moving away from the hotspot, and as they do, they erode down and become smaller. So the big thing, though, is that this system allows you to look at different stages of diversification. And so you can see different stages of how the overall evolutionary history and how the communities come together over time. And just to illustrate this, if you look at what the Hawaiian Islands looked like 5 million years ago, the these islands actually were fairly low and far apart, but when Kauai formed about 5 million years ago, it was really pretty much on its own there. These islands, as I said, were low and far apart. But so then you got to Kauai that was largely colonized from outside. And then Kauai, the organisms that reached Kauai would have hopped down to Oahu. And then you got Maui forming, and they would have hopped down to Oahu, to Maui, sorry. And then you get the Big Island forming here, and they would have hopped down to the Big Island. And then this is what you see now. And so basically you can say, oh, well, here you have a community of organisms that is 5 million years old. And this is one that's kind of 2.5 to 3.5, and this is around about 1.5 million years, and the youngest one here is 500,000 years. So basically, what this allows you to do then is to read the island sequence kind of as the fossil record, and that allows you to look at the dynamics of trait evolution and species diversity through time, and the temporal dynamics in niche interactions, so how the whole community changes over that time. So how has this been examined so far? And the insights into how you might be able to use this system rest fairly fundamentally on really a piece of work that was developed by Warren Wagner and Vicky Funk in 1995. They put together this edited volume here on Hawaiian biogeography. And what they did was they just got together the people who were then just starting to work in the Hawaiian systems. And it was really the first attempt to test the idea that independently derived groups of Hawaiian organisms exhibit similar patterns of colonization and differentiation. And the thing is, you want to have these similar patterns of diversification so that you can look at how whole communities come together. So I'll just think about the progression rule first of all, and just see what are the nuances about that. So here you have the Hawaiian Islands, and the idea behind the progression rule is that organisms simply go from the oldest to the younger islands. And this might make sense in many ways because you have a new island forming, and the only place they can come from is the next most proximate area, at least the most likely place that they're going to come from. And the idea is that, you know, once an island is filled, so to speak, then things can't go back, and so they tend to go in a unidirectional manner. And this is actually this is what a lot of organisms do. So they tend to hop down the island chain, although they can also differentiate within islands. So I'm going to use this as a kind of backdrop to look at three different things. Look at the evolutionary dynamics over time, so looking at the predictability of the evolutionary process in different groups. And then looking at overall species diversity through time, so how communities come together over time. And then try and use the island's temporal sequence to go from microevolution to macroevolution, looking at the interaction of isolation and how interactions change over time. So just let's first look then at the evolutionary dynamics within lineages. So we'll just see how different lineages really perceive this chronosequence. And so here you have, just as an example, I'm going to give you a few examples here. These are bonza crickets. And you can see here that you have the oldest taxon on actually one of the atolls right up to the north. And they basically show this progression down from the oldest to the youngest islands. So this is a fairly common pattern. And here you see Clermontia, and again here, by and large, they've gone from the blue to the red. And so mostly progressed in this direction towards the younger islands. There's almost always one or two in these various lineages, one or two that go back in the reverse direction. But predominantly you get this one-directional colonization. And I should say, though, that the organisms that tend to show this progression are the ones that have been in the islands for at least 5 million years, so that you can use the island sequence as a chronology, so that they've arrived at the oldest island and then they tend to hop down. I should say that it is now considered that most organisms in Hawaii are 5 million years old or less. And this was the work of Jonathan Price and David Clegg. They just looked to see how old different lineages were, and it seems that most lineages have arrived more recently than the very old islands. But although there's still some that are younger, and there's still a few that are older. But by and large, though, you get this progression going from older to younger islands. So here you see the two different plant lineages, Shidea and Psychotria. And you see they go basically, here's the Kauai and Kauai here, and they tend to go in the direction of the younger islands. And so there's deviations, but I mean there's some back colonization, but the progression is the overall picture. This is a picture from a slide from the work of Kerry Shaw on Laupala crickets. And here again, they show this beautiful progression. Two different lineages, but the oldest one is Kauai, and then you've got two that have gone from Oahu down to the youngest island. So it is a very dominant pattern. And so you get this, what's interesting is you get this even in taxa that don't differentiate tremendously. This is actually the Hawaiian happy face spider, and this is work that Pete Croucher, a postdoc in my lab, did. And so this is based on AFLP data, but the point here is that they're very discrete. They're a single species throughout all of the islands from Oahu down to the Big Island, and they're very discrete populations. But what's interesting is you get, I mean, even on the Big Island, you get very discrete entities, and they've still followed this progression from the older to the younger islands. So what this is showing us then is whether or not lineages diversify, you get a progression down the island chain. So this is a general pattern for those that colonized on the oldest islands or before. And this is often accompanied by rapid speciation. Okay, so we've got that kind of as a context, that many of these groups show a progression. And so you can kind of get this temporal series. You can use the islands as a temporal series. So then what is, I mean, do they all show the same thing? Or is there, is it more nuanced? And this is where it starts to get more interesting. So what I'm going to do is look at the pattern of diversification and in different groups. And so what's interesting here is that you've got this, in different groups, you have a balance between geographic and ecological isolation. And so here we'll start off with a snail group. And I worry about talking about snails with Dermot here, but I'll tell you a little bit about what I know of snails on Oahu. So snails have diversified prolifically, and really beautiful snails in Hawaii. But what's interesting about the snails is that it's a so-called non-adaptive radiation. And so they progress by and large from the older, this group is not on Kauai, but they by and large progress down the island chain. But they're allopatric, so they don't actually co-occur at a given site. And so that's why it's considered a non-adaptive radiation. But there's a number of groups that do this. So we'll get back into familiar territory with the spiders. And this is work that I did with Gustavo Hormiga and Mikel Arnedo. And we looked at, again, multiple molecular markers. I should say that most of what I'm talking about is based on various kinds of molecular markers. But this is work we did a while ago based on sequence data. And this spider is the genus Orson Welles. Again, Gustavo described this one. He liked Orson Welles, and you'll see that all the species are named for different Orson Welles plays. But the point here is that they do show this progression from, they go from older to younger islands. But what's really interesting is that if you start looking from kind of using the chronosequence in the other direction, so going from the youngest to the oldest island, what you see is here you've got one species on the youngest island, and then you go a little bit older, and you've got 3 species there. 3 species on Oahu, even though it's so much smaller, and then you've got a whole 6 species on Kauai, so they've just built up. So what seems to be happening here is that you've got a widespread species on the youngest island. It just hasn't really had time to do much. And then over time, they just diversify without changing ecologically. So it seems here that you get diversification over time. It's non-adaptive, and it occurs in allopatry. These species don't co-occur. So that's one pattern, and a number of groups do this. However, of course, as you know in other groups you've heard about, there are other patterns. And here I'm going to show you some fairly recent work that's come out of a really huge lineage of Microlepidoptera. These are little Lepidoptera. It's a very large genus, Hyposmacoma. And this is work by Dan Rubinoff and people in his lab. And what they found is that these moths diversified into very discrete ecological forms early on in the radiation. So you get early ecological differentiation into these very discrete forms. And then within each ecological form, they show this pattern of going from oldest to youngest island. So early ecological differentiation, and then subsequently hopping down the island chain. This is a pattern you find in a number of groups again. And this is a crab spider in the work that Jessica Garb did in my lab. And what Jessica showed was that, again, you get early ecological differentiation into the most distinct taxa. And again, based on molecular data. So all the blues are on the oldest island. And you can see it best if you look here. Here's the blue one that's formed here. These are all different species. But then what happened is they just went down to the younger island, and then the younger island, and the youngest island. So diversified ecologically on the oldest island, and then hop down the island chain. So here you get adaptive diversification, and then they diverge subsequently in allopatry by simply hopping down the island chain. So here, this is just to show you very early ecological differentiation, and then they just hop down the island chain from the older to the younger islands. So adaptive populations or species differentiating in allopatry on different islands. Now, that's all fine and well, and we say, okay, that's another pattern, but there are others again, and you're also well aware of that. And so these are actually the really interesting ones. So in some groups, they've hopped down the island chain, but diversified within islands. And I'll give you just one example here. These are Hawaiian violets, where they do that really nicely, and you can see very clearly how you get repeated evolution of similar forms. The blue form is bog, the green is swamp, but they show repeated evolution of the same forms in multiple different groups. Actually, silver swords show that pattern as well. But we'll come back to spiders, where I'm more familiar. And here I'm going to show you just a few stories here. So these are Ariamne spiders, and this again is sequence data, and it's preliminary data. But what we've found is, based on sequence data that we have, they've basically hopped down the island chain from the older to the younger islands. And the thing is, within that context, they've diversified within each island. So you get the hopping down the island chain. But what's happened then, if you look at each of the islands, you've got a gold one popping up over and over again. And you've got, so this is the gold one. The gold one lives under leaves. And then you've got a brown one popping up over and over again. And this is the brown one that lives in rocks and things. There's also a white one. This is the white one here, and it's actually, we've found a white one on Oahu too. So you get the same forms popping up over and over again. So that's Ariamnes. But there's other groups that do this. This is the group that I've worked on most. These are Longjawed spiders, the genus Tetragnatha. And they've diversified all over the place. They're extremely diverse and narrowly endemic to specific sites. You get a whole bunch of them that spin webs, and then you get a whole bunch of them that don't spin webs. Basically, they do this, what all spiders do, but they're just a single genus. They've done this classic adaptive radiation into multiple different ecological roles. So when you look at how they've diversified, one of the major foci of what I've been doing is in the spiny leg group that don't spin webs. And in the spiny leg group, you get these distinct ecomorphs. The brown one, which is here, so the large brown one that just sits on tree trunks and slow and docile and heavy. You get the green one that runs around leaves, the maroon one that's on mosses, and the little brown one that's on twigs. So discrete ecomorphs, they've got the ecological affinities, they've got morphological affinities. And what you find is that when you look at this using molecular data, what you find is that they've repeatedly evolved the same form. So here you see just molecular phylogeny, and you see the green ones popping out over and over again, not monophyletic at all. The maroon ones, they pop out, and the big brown ones likewise. So you get independent evolution of ecomorphs. And you say, okay, well, that's great, you know, for these spiders. I'll just show you one more here, and this one is quite interesting as well. What do the web spinners do? So here are the web spinners, and so they exhibit all sorts of different kinds of web structure, and this was work Todd Blackledge did in my lab, and he looked at the web structure, and this is a principal component analysis just to look at the web form. And so within a habitat, he looked at the web measurements in this principal components kind of framework, and he found that within a habitat, they're very distinct. So this is Oahu, this is Big Island, this is Maui, and you put them all together, there's a lot of overlap. But when you look at that in a phylogenetic context, what's cool is that they're not, you know, the ones with similar web types are not at all closely related. So basically, you get repeated evolution again of the same web form. Even though in this case, the spiders have gone, I mean, they show no convergence at all. They've done whatever they wanted. So here again, you get evolutionary convergence and repeated evolution of similar forms. So basically, what you're finding here is that on the oldest island, then you get one, you know, you would have got something arrive, and they diversified ecologically, and then they hop to a new island and diversify again, and the same, a couple maybe to the next island, diversify again. And so you get this pattern of repeated ecological diversification that's adaptive, and you get within island diversification. So what we're seeing here then is that you've got a balance between diversification, between geographic and ecological diversification. And this is what seems to dictate the pattern of diversification. So on the oldest island, this is where you would have had, this would have been extremely isolated when organisms got there, with all the older islands pretty much low and far apart. And so there would have been more time for adaptive exploration. And so it seems that that could explain why you get so much more, in many more groups, you get adaptive exploration on the oldest island. And then as new islands form, you get propagules arriving. In some cases, they just pop down, and colonists just from the oldest island pop down and don't do the same, just keep doing what they were doing on the previous island ecologically. But then in other cases, they hop down, and then they colonize and then adapt quickly into another ecological form. So what's cool about this is that it allows you to look at different stages of the whole diversification process. So you say, okay, this is, you can look at Kauai, look at radiation, and just look at Kauai. And here's what you often see on Kauai. Long branches going to your terminals, and you say, can you reconstruct the ancestors and the ecology based on this? Can you say what was going on in the history of this lineage as it was evolving based on that information? And it's hard. I mean, that's what a lot of us are stuck with when we're trying to look at a lineage and reconstruct what's happened in the past. So what we can do, though, in Hawaii is that you can say, okay, that's a 5 million year old lineage, but you've also got it on the younger island. And so you say, well, this is what it was doing 3 million years ago. And so you can say that this is a stage that's going to go to this. And then when you go to the younger islands, often there's just tremendous diversification. And you can say, well, this is the earlier stage, and it'll end up like this, and it'll end up like this. And what's also really interesting is that on the youngest island, you often don't get monophyly. And the question is that because they will hybridize and end up in monophyly, or is it really that the youngest island is different? So at the moment, you often, in many groups, you just get populations from other lineages having got to the youngest island. So what we can do is look at patterns of diversity now. What I want to do is look at patterns of diversity across the entire archipelago. So let's just think about how that works then. So what we're going to do is look across the entire sequence. So you can see we've got the progression rule. Now, there has been work to try and figure out what goes on across the island sequence, and this is work that Peter Vitusek did. And Peter is a soil ecologist, geochemist, ecosystem biologist. And Peter was interested in how ecosystems change across the island chronology. And so he established this long substrate age gradient. And based on this, what he found was that you get very distinct changes in the soil properties and associated ecosystem properties as you go across the chronosequence. So you get very low nitrogen and phosphorus very early in the chronosequence, very low late on, and high and peaks in the middle. Susan Kennedy, who's a student in my lab, has been looking at this to see whether these soil properties are actually reflected in the organisms that live there. And what's actually quite cool is that she found that on the very young substrates, here you've got the nitrogen isotopes on the very young substrate, and you see that leaves have the lowest nitrogen. So this is the stable isotope of nitrogen. You see that the heavier isotope is it increases as you go up the trophic level from these are the leaves. These are spiny-legged spiders, non-web building, web building, and spider-eating spiders. And you'd expect this. You go up the trophic level and you get an increased amount of the heavier isotope of nitrogen. But what's really cool is that you go to the older site and it reflects what the soil's doing. So you go to the older site and they ratchet up so that you get the same patterns, but they've ratcheted up the amount of nitrogen, and likewise the older sites. So you do, what's important here is that the organisms that live at these sites are reflecting what the soil is doing. So they are reflecting the geological age of the environment. So now let's just look at overall patterns of diversity across the archipelago. And here's what's really quite interesting. So this is looking at all plants that have been in the islands for at least the age of the current oldest island, so Kauai. So given that plants have been there for that length of time, what are the patterns of diversity? This is straight species per unit area. And what's interesting is that the species per unit area in many of the larger groups increases really quite a lot, and then drops off or levels off on the older islands. But in some of the smaller groups, it keeps plugging along. And what's interesting here is that you find the same pattern in animals as well. The very large groups seem to show these peaks, and then they either drop off or level off, and then some of the smaller groups just keep plugging along. And what makes this, you know, if you think, well, what in the world's going on? Let's come back to spiders, where I know a little bit more. If you look at spiders, which spiders are doing which kind of thing? Oops, sorry, just a little glitch there. So which spiders are doing which thing? So here's Orson Welles. Orson Welles, remember, non-adaptive radiation, and it's just what it does is the highest number of species on the oldest island. So Mecaphysa, which is a crab spider that has the most ecological differentiation on the oldest island and then hops down the island chain. Here you've got a very rapid accumulation of species, and they just kind of level off. And here you've got the two groups that show this rampant early differentiation where they all come together in sympatry very quickly. And here they've got by far the highest diversity on these very youngish islands, not the youngest, but the younger island. And so if you look at this overall, this is just a very basic pattern, but what you see here, this is the Orson Welles just plugging along and with much the highest diversity on the oldest island, and then Mecaphysa, and then these ones here, the ones that proliferate and come back together in sympatry, they show these kind of peaks of diversity and then level off. So it seems that in these ones, it's not entirely clear. We don't have enough data to say how real that is or what might explain it. But the bottom line is that what we know is that diversity is low for all species groups on the youngest island. And it keeps increasing in some of the groups, in particular smaller groups. And then in these other groups, it rises quickly and then levels off or declines. And these are often the taxa that quickly come together to co-occur in sympatry. So this is something we're trying to understand. And so the question then is, can we detect signatures of these differences on the youngest island? So this is when, you know, we've been talking about across the entire island. And so what I want to do now is start looking not just at the entire chain. I want to come down and look at the younger areas. So we know that there are very few species on this very young island. We know that on Maui that things have really got going, and there's a lot of different species there. So what might be going on here? The first thing we wanted to look at was really on this young island, the big island of Hawaii. It's got 5 volcanoes on it. And so you say, well, how have things diversified on that volcano? And so you say, well, maybe, you know, they started on the old volcano and then went to you know, as the new volcanoes popped up, they're all different ages. So, and some of these ones, this is Mauna Loa just building, and Kilauea, again, just building. So did they just simply hop down the volcanoes? And so do you get signatures of this just hopping down from the older to the younger volcanoes? I mean, that's certainly something that might be going on. Another thing that might be going on is that you get, they get to the new island and then just expand ecologically and cover the entire area with an expanded ecological amplitude. So that could also be going on. So what might be going on? So let's have a look at some of the populations then we've looked at on these young islands. And here we come back to the Hawaiian happy face spider. And as I said, you know, this spider here it's all a single species. It has this balanced color polymorphism. This is actually a male guarding a female, and the male is actually he isn't really brown. He is brown, but he's just eaten something, and that makes him brown. But they're usually pale yellow, translucent. And the most common form is yellow, and then all the rest of these different colors. But the point is that it's a balanced color polymorphism within a given population, and all of them have this. But the point is that they occur right across the islands from Oahu down to the Big Island, and they're very discrete populations on each island. And so even within Hawaii Island, you've got discrete populations. So, but the thing is, it's not entirely that, because what you see is that especially when you get to the youngest volcano, you get a lot of mixing, and you can say, well, you know, that's expected because it's super young. And so we've been looking at these structures in different groups, and this is data that's just coming out now, and this is work that Ellie Armstrong and Stefan Prost have just done. And this is using RAD-seq data, DD-RAD, and looking at populations of the Ariamnes. And here, these are older islands. This is the brown species. It's actually all the gold, except this is the brown species, Ariamnes hiva. So these ones are all Ariamnes waikula, all on the Big Island. And you see, by and large, they separate out in discrete populations, but that's not entirely the case. And so let's look at this a little bit more clearly. And what you see is that this is work that Kari Goodman did in working in our lab. And this is plant hoppers. And what Kari showed was that you get very discrete populations. These are the east, this is Mauna Loa, they're blue, and Mauna Kea in the purple. And so you get discrete populations, but all the ones with the dots are actually mixtures of different populations. And what's interesting, you get the biggest mixtures are actually in between Mauna Loa and Mauna Kea. So let's have a look at what's going on there, and this is quite cool. So here you've got what's going on in that area. And what you see, this is Mauna Loa, and the red is the lava. And so what happens is the lava carves out the habitat, and it's continually fragmenting that habitat. And so what you find, this is again happy face spiders, and you know, you might expect, you know, discrete populations with large genetic distances, but this is just a mitochondrial DNA haplotype network. But the point is that in the light green are the kapukas, these little sites between the two volcanoes, and you get a lot of haplotype diversity. And we're exploring this further at the moment, but I'll just give you one little snippet of what might be going on here. And this is, again, looking at the youngest island and looking at this one area here between the two volcanoes. And here is where you get, if you look at this one site here, what you find is the gray is the forest, and the white is the lava flow. And it's recent, 1855. And I had a graduate student a while ago, Amy Vandergast, and what Amy did was she looked at spiders inside these little islands of vegetation. See, this is outside the kapuka, this little island of vegetation. This is outside, this is inside. So a dramatic difference. So they're really islands. And you get some species that are confined to these little islands, and some are outside. And outside and on the edge. And what she did was she used mitochondrial DNA analyses. This was a while ago. But she looked at isolation by distance, in particular, where the forest fragments were surrounded by lava, which is shown by the red here. And what she saw was that you get very marked isolation by distance when these kapukas are surrounded by lava, and not when they're continuous forest. So does the lava limit the movement? It certainly does. And you get very different compositions in these different forest islands. And so what exactly is going on in these areas? And this is what's really, I find really exciting, and work that we're doing at the moment. This is something that Hamp Carson talked about a while ago, And he commented on the significant differences between even closely contiguous kipukas, these little islands. Huge genetic diversity retained, likely a result of the shifting mosaic of the landscape. And this is something that both Hamp Carson and he's talking here to Peter Grant, and they both come up with this idea. So Hamp said that founder events may play a role in just the repeated extinction recolonization of these habitats. And basically, it's allowing different populations to come together, they merge and add to the diversity. And this is something that Peter has been talking about as well, that the birds exist in a kind of equilibrium state, dying and replenishing, more or less equal rate over the long term. This fusion and fission oscillation goes along a lot in biological systems. So then what we're thinking here is that you've got a balance between isolation and time. The isolation separates populations and the fusion injects high diversity. So the idea then is you've got these pockets that get isolated and then they come back together and potentially infuse genetic diversity into the system. This is now known in cichlid fish. What Peter was talking about was it occurs in birds, and what we want to do is get a handle on it in the arthropods. So just I'll go through this really quickly just to show you how this might play out over time. What we've looked at, again, very preliminary data, and looking at isolation of these different populations of species and how it plays out over time. And this is just the NEM between different populations. And you see that, and these are the FST values in the blue. And the point is that you get between different volcanoes, you get much higher genetic distance on the older islands compared to the younger islands. And this is true whether you have speciation or not. So in the top two, you get speciation. In the bottom, this is the happy face spider. But again, much higher divergence between volcanoes on older islands compared to younger islands. So with speciation or without. So just quickly then, what exactly is going on? And I want to tell you just a little bit here about what a graduate student, Darko Kotaris, did in my lab just recently. He used exon capture data to look at this problem. Here, you know, we talked about Tetragnatha and how you get the highest diversity on this island here, the Maui. And why do you get, 3 species of the green taxon are on the same volcano? What in the world's going on there? And so what Darko did was look at these populations, and what he found based on the exon capture data was that this one species, Tetragnatha waikamai, likely established on this one volcano and probably expanded everywhere to Lanai throughout the Maui Nui complex and over to the Big Island. And then based on this data, what it seems was that we're still working on this at the moment, I should say, trying to see how lineage expanded just to get the directionality here. But it seems that what's likely happened is they changed on Lanai to different species and on the Big Island, and then all came back together on Maui. And so that you get these multiple species right together on the same island at the moment. So what we can say then is that geographic isolation plays a major role in the early diversification of all these taxa, though isolation imposed varies by taxon, and it's super dynamic. So here I just want to introduce more of what Andy will be picking up on later. We need to understand the properties of the communities overall that can provide a signature of what is different and novel and how they differ. So what a big project that we're doing at the moment is a Hawaii Dimensions Biodiversity effort, and we're looking at sites of similar elevation, precipitation. They've all got metrosideros forest, and they've all got reserves. So basically what we're doing is trying to control for elevation, for precipitation. They've all got forest, and they all have reserves, but what we're doing is changing the geological age of the system. And what we're doing is we're asking how to, oh dear, never mind. Oops, we don't want to send that information to them. So what we're doing then is trying to see then how the entire community changes over this sequence. And just a sec here. We won't send it. And we'll, oops. It saved how many temporary parts of this? Let's see. Well, it just has to do its thing. So just 2 shakes. So here we go. Oops, here. Oops, sorry about that. So we'll go down here. So what we're doing then, and if we just, the anticipation. So what we're trying to do, if it'll get its act together here. So what we're trying to do is get together a lot of ecological data based on multiple different methods. So we're using lots of this. This is canopy malaise. This is beating. Oops, sorry. And then I pressed the forward button just when it got exciting. Just to show you, it's probably going to have a conniption there. But we're getting all of this ecological data from all of these different methods. And so the point here is that across the chrono sequence then, we're getting a lot of ecological data that can allow us to address really fine, detailed information about how the characteristics of the community is changing over time. And just to show you what Andy will talk to you about the data you can use, you can apply for generating ecological metrics across the sequence. But the thing that we can get at really quite nicely through this approach is DNA sequence data for the entire community. And this is work that Henrik Krehenwinkel is doing in our lab, and he's getting information, DNA data from the entire communities that we're getting from these messed up samples. And he's working on this at the moment, but what you can just kind of grind up the whole thing and get information on the entire community composition. And this allows you to look at, hopefully, at exactly what you've got there, even for cryptic taxa, relative abundances. And what it can also do is allow you to get at interactions between taxa and looking at arthropods, looking at herbivores and their plants or predators and their prey. And so what we're hoping to do then is use this to look at basically how communities are changing over this island sequence. So just to sum up then, what I've hoped to show you here is that we can use this data then to look at which taxa first colonize areas, in what order do they colonize, is it predators, prey, what are the interactions in those early stages of differentiation, and in what order does this relate to early trait differentiation, in what order do these attributes together play into adaptive radiation and the development of biodiversity. So overall then, what I've tried to show you here is just how you can use the dynamic of the Hawaiian Islands to look at evolutionary dynamics over time. So looking at the repetition of the evolutionary process and how different arthropod groups as well as other groups show different patterns of diversification. They seem to perceive, colonize, and diverge differently across this evolving landscape. And looking at species diversity through time, we've looked at how communities come together and how patterns of species accumulation differ between taxa. We've also looked at microevolution to macroevolution and how isolation and admixture play a role in fostering the initial diversification, but it differs depending on the taxon. And we've also just introduced the idea of how you can use ecological metrics of abundance and interactions to allow assessment of ecological shifts over evolutionary time. So that's all I want to tell you, except just to say that to emphasize here that we've been looking at an entirely native system, a super isolated system. And just so that you understand that I'm not entirely living in a bubble and don't know what's going on, Hawaii is no longer isolated. And so you've got a system here that has basically been been smooshed together. You've got, the Pacific is effectively shrunk, and you've got a lot of invasion going on. And so as a next step, what we want to do is figure out how the native dynamic is affected by this new dynamic. So just for example, here you see a native system here on the right, invaded system on the left. This is the Nature Conservancy property. This is ginger that's just coming into the Nature Conservancy property. It hasn't got in. The fence is actually not keeping the ginger out. It's the people that are trying to keep it out of the reserve, but you can kind of see that this is an uphill battle, and this is something that we really want to see how this dynamic might play out. So with that, I'll end by just saying there's been a lot of people that have been involved in this, and yes, it's been great talking to you, and I would love any questions that you might have this time. And yes.
So in your first part, you talked about a lot of the very different patterns by different groups of species. Are there any consistencies in which taxa show which patterns?
That is super interesting, isn't it? So some taxa just don't do the adaptive thing at all, and some do it a little bit. So which ones do what? And we really need genomic data to get a handle on that, and that's what we're trying to do. But is that going to provide an answer? We can be pretty sure that it's that for the ones that keep switching, there's kind of a predisposition. It seems really easy for them to keep switching to a new suite of traits. You know, once they've done it once, they can do it again. But we don't. Could we predict which ones are which? No.
So not based on any ecological traits, trophic position, kind of predator mode?
Nothing, as far as I can see. Andy might be able to say something different, but there's nothing there. I've been focusing on spiders, and as you can see, spiders are tremendously homogeneous in terms of the kinds of attributes. I mean, they're all predators, by and large, and so they do the whole gamut. So it's a fascinating question there. Yes.
With the group of taxa that lose diversity on the older islands, do you think the erosion and the loss of habitat has something to do with that group?
So for sure, it has something to do with it. The loss of area, the habitat diversity itself has. On the oldest island, it's still pretty diverse in terms of the kinds of habitats they have. But why one group shows one pattern and, why some just keep plugging along? It's really the difference between the groups that is not at all clear, Yes.
Please join me in thanking Rosemary.
Okay, thank you so much.